So before we continue with the presentation, let me introduce to our guest speaker, Dr. Stefano Vanin. So Dr. Vanin has been actively involved in research and teaching since 1997 after having been awarded a Master's in Biological Sciences at the University of Padua in Italy. So in 2001, Dr. Vanin has been awarded the PhD after some periods of research in France and also in the United States. After 9 years at the University of Huddersfield in United Kingdom as a reader in forensic biology, Dr. Vanin has moved back to Italy as a professor at the University of Genoa. He has focused his research on the behavioral, neuronal, and also the physiological adaptations of insects to temperature and light. Also, Dr. Vanin has researches on forensic entomology and also on funerary archaeo-entomology topics. So, for example, species distribution, insect colonization in burnt and buried remains, the effects of globalization and global warming on forensic imported species, and also the molecular identification of present and also archaeological specimens. Dr. Vanin's publication list counts more than 120 publications, including books, book chapters, and also articles in both national and international journals. In 2015, Dr. Vanin organized at the University of Huddersfield in UK the first international conference in funerary archaeo-entomology and also the 12th international meeting of forensic entomology under the umbrella of the European Association of Forensic Entomology, also known as EAFE. In 2017, he also organized the second international conference in funerary archaeo-entomology and the 14th international meeting of forensic entomology in Treviso, Italy. Dr. Vanin is a fellow and also a member of several national and international associations and societies, and he is also currently the president of the European Association for Forensic Entomology and also the Italian Association for Forensic Entomology. So with that being said, I cordially invite Dr. Vanin to begin his presentation. The floor is yours, Doctor.
So first of all, thank you indeed for this fantastic invitation, and I hope to come to your university in the future when this horrible nightmare pandemic will be stopped. And I think that we have the responsibility as a single individual of this earth and member of the humanity to fight all together against this virus. So really, I'm happy and honored to be with you as well in a virtual mode. But technology is helping, so we need to use whatever the time in which we live offer to us. So I'm really happy that in this case, technology is able to shoot down and cut the distances. Just 1 second, because a few seconds ago, I was able to share my screen. Now, et voila, sim working. Could you please confirm me that I'm sharing the screen? No?
Yeah.
Yes.
Yes. Okay, great. So as I've been introduced, my talk, sorry, has been introduced, I will keep you on the hands, and I want to walk with you through the past, but in a quite different way. We are used to read historical books, we are used to listen or watch a documentary about the past, and I want to use, in contrast, insects, bugs and flies to try to understand what's happened in the past. And you know, insects are quite important from this point of view. But before starting, I know that I've been introduced from an academic point of view. I want to introduce myself from a personal point of view. So, and I decided to do showing my heroes, the people that were my heroes when I was a young student. And I think that some of you are able to recognize among them some friendly face. Here we have a group of naturalists from Darwin to Coral Lawrence, the father of ethology. We have Wilson, who created the word biodiversity, and then other one. Then another group of writers, but not only writers, they were as well traveler, people that were used to fly around the world or walking around the world. And last but not least, for my personal education and development, these three guys, an English, an Italian, a French, belonging to the Boy Scout movement. So this is my background. And all these people are really at the base of my curiosity, my enthusiasm, and my willing of going around the world to discover new things. I'm not able to stay in the same place for a too long time. So because of these people, I start being interested in entomology. So insects were my passion, and I did my first dissertation on the life on the glaciers. And on the glacier, I met glacier of the Italian Alps, of course. And then I will show you where Italy is and what Italy is. Glacier of the Alps. And then in that place, I met the fantastic world of flies. Because my supervisor, husband, was a legal doctor, a forensic pathologist, my supervisor invited me to join him to start studying flies from cadaver. And this is the reason why I become initially a forensic entomologist. Of course, I'm speaking in a group of entomologists, of people interested in entomology, so I don't have to spend too much time explaining that insects are the most important taxon among the animals. There are 73-75% of the species, and they are characterized by an exoskeleton, and for this reason they have to change the skin, the cuticle, to be precise, to grow up, and they were able to colonize all the environment just because they have wings. And I like this painting that I found in some gallery in a museum, because as you can see here, as well, the angel, from the Christianity point of view, they copy the wings from the insect, or maybe the insect they copy from the angel. I don't know the direction, but it's quite interesting because looking at the painting in the museum, we are able to recognize as well the butterflies from where that have been used by the painter as a model. And this is amazing. So I suggest you, when you go through the museum, switch on your attention, increase your attention in the small detail, and you will be able to see insect or part of insect as well, where they are not supposed to be. But anyway, just to refresh your memory, the cycle of the insect, or better, some of the insect, the Holometabolus, go through some stages. We start from the adults, they mate, sometimes they do not, because females are able by parthenogenesis to reproduce without the males. Anyway, we have eggs. Sometimes we don't have the eggs because the female are able to larviposit, this means lay directly the larvae. Then we have different instar in the larva development. Then we have the pupariation. Inside the puparium, we have the pupa, metamorphosis, and then we go back to the adults. We have this for flies. We have the same for beetles. What is important is, and I ask you to pay attention to this point, is that puparium, this cocoon, is resistant and strong. The adult is not so strong. So in an archaeological context, in a past context, because could be as well a forensic case, an older forensic case, what we can find associated with the body for flies are puparia. Instead, for beetles, in the majority of the case, we will be able to find the adults, elytra, part of the legs, sometime, but only sometime, the head of the larva, because is the strongest part, and for their mastids, we could find as well the last segment of the larvae with the specific horns. So I'm pointing out the attention on the fact that for flies, we will never find the adults, but we will be able to find the puparia. For beetles, we will be able to find other developmental stages. But as entomology, I think that we can give a diploma to everybody in entomology if these 3 points are memorized. So insects are a lot, but they are different. So they have different biologies. That means they feed on different kind of food. They develop with different times. They have different mating or reproductive system. Second point, the insects are everywhere, but they have a specificity of habitat. So I have some insects that are typical of the coastal area, and other one that are typical of the mountain. Some are typical of the urban environment, and other one, the asynanthropic, they live in the countryside. Then we have to study in a very precise way the landscape to understand if this difference is evident where you live. But where urban and agricultural environment are separate, of course, we can see insects belonging to different group in different places. Last but not least, insects are always present, but have a specific phenology. If you prefer, you can speak about seasonality, but I prefer to speak about phenology. This means some are active during the winter, others are active during the summer, of course, depending on the environmental condition of the country where you live. But what is archaeo-entomology? I start with this general definition of archaeo-entomology, and then I will go through funerary archaeo-entomology. Anyway, archaeo-entomology, as defined in 1994, is the study of insects collected from archaeological deposits to reconstruct their cultural and natural environment at the time of deposition. So be careful, because sometimes people mistake forensic with whatever, and we will see later with funerary archaeo-entomology. But what forensic entomology, archaeo-entomology, and more or less all the disciplines that work using evidence collected nowadays to understand what's happened in the past, they share a word. And this word is reconstruction. We study insects to reconstruct what's happened in the past. We were not present at that time. We were not present for forensic entomology on the crime scene. But looking at the insect that I'm able to collect now, I can reconstruct the past. So forensic entomology, archaeo-entomology, funerary archaeo-entomologists, they try to answer the question about when, where, what, how, and sometime who. Of course, we will be not able, never, to answer on the question why. But if we focus the attention on archaeo-entomology, we can see that it's useful to reconstruct the environment. Because if in an environment of savanna, nowadays savanna, I found some species that are typical of a snowy environment, of course, it's telling us, the insects are telling us that in the past, that environment was not so warm and dry as it is now. So first point, we can reconstruct the environment, and we can reconstruct as well the climate. This is done in a very, very precise way, studying as well the chironomids, the midge, head, capsule head of the larvae that we can find at the bottom of the lakes. Studying the column, of course, of sediment, we have a sort of stratification, of course, of the deposit in the lakes that include as well the head of the midge larva, of course. We will be able to identify the species, and because the specificity in terms of temperature resistance, we can really understand what's happened in the past in terms of temperature and environment. But then, and of course, not only with the midges, we can do this one as well with beetles, because as I told you a few minutes ago, they are particularly resistant. Then insects tell us about biogeography and the way in which people move around the world. There is a fantastic study about the association between human and lice. The way in which the lice, they move around the world, that is associated with the human movement. And another thing that is amazing is related with the reconstruction of agricultural and storage practices. So Roman people spread pest insect associated with some vegetable around Africa, and we can, and Dr. Patanjopoulos produced a fantastic paper. To be honest, she did her PhD thesis on this topic, demonstrating the way in which the Roman moved through Africa using the deposit of pest insect coming from Europe. Of course, in terms of archaeology, we can use insect to understand as well the health, the hygienic condition, and this is the domain of archaeoparasitology. And of course, we can use the insect as well in this big new discipline that is called Funerary Archaeo-entomology to understand the funerary practices of the past. Why we are so interested in the past? I think for two main reasons. One, because we need to know our roots and the way in which our thinking and our society built up. Really, we are related with the past. The second one, because Latin people were used to say storia magistra vitae. That means history is a teacher of life. I would like to add, unfortunately, she doesn't have too much students because looking at what is going on in the last year, this means that we forgot the lesson of the history. But anyway, we can learn a lot looking at what's happened in the past, in terms as well of health, but not only health. But what is funerary archaeo-entomology? First of all, I would like to introduce you my friend, Jean-Bernard Rouchet, that is a French researcher. And in this picture is me and him at his house. This is the dinner that, this is the appetizer that he organized for me when we had this fantastic evening. And he's a crazy guy. As you can see, he put some olives on plastic ants just to create a nice environment between and among entomologists. But Bernard Rouchet had this fantastic idea to apply the knowledge and the technique that we use in forensic entomology to investigate human bodies, and not only human bodies, because we will see later, it's not related with only with human bodies, to use at this point insect to reconstruct what's happened to these people. So if you look at the definition he gave in 1996, we put attention that is the application of forensic entomology in funerary remains. of archaeological interest. So please, this is a point that is very, very important from a theoretical and epistemological point of view. Forensic entomology deal with something that is forensic. That means we have a mandate from a magistrate, from a court of law or whatever, or from the police to investigate about a forensic case. When we apply INSET in the past, we are not doing forensic entomology because it's not forensic anymore. We are doing funerary archaeo-entomology. So please, as well, in your future paper, do not use the word forensic entomology for archaeological case. In one case, there is a mandate, there is a law. In the other case, we want to investigate about the past. And forensic doesn't mean more precise. The application of the scientific method is already the base, the bulk of our precision, our accuracy. We are scientists. Then we can do something forensic, but it's only because it's related with the court. So, as you can know, some insects are specialized in the consumption of decaying matters, which include any sort of dead tissue from animals and for humans. It's quite, I would like to, sorry, when I wrote, and I'm used to write this sentence, I have this sort of conflict in me, because from humans and animals sound strange to me, because humans are animals. So from a biological point of view, we are made of the same substance. Of course, there are some differences, and we will see later which one. Or you know better than me what is the difference between our species and the other one. But for insect, we are not different compared to a cow, a pig, a dog, a cat, a fish, or whatever you want. So in terms of the composition, we are flesh, we are energy and matter that arrive in a system. And you know very well that insects are able to completely destroy the organic matter. And we need, from an ecological point of view, this, because if not, we will have carrion and cadaver everywhere, and it would be impossible to live. I know that before me, someone already told you about forensic entomology, so you are already confident with the main group, the main taxa that are able to colonize a body. They are Diptera, flies. Here on the bottom, Lucilia. On the top, species in the genus Sarcophaga. Remember, please, that Sarcophaga doesn't lay eggs, it lay directly larvae. On the right, on the bottom, you have a clothes moth, and on the top, a beetles, a larder beetles that is quite common in Europe and is affecting not only the cadaver, but as well the food storage. You know that during the decomposition, a body goes through some stages, from fresh to skeletonization, in specific condition, very dry environment. That doesn't mean warm, because we could have very dry environment in cold places. On the glaciers, we have mummification, as we have in the desert. So we have the decomposition with the putrefaction and skeletonization in mild environment. When the environment is dry, could be cold, could be warm, we go through the mummification. Sometimes a body could go through the two situation. We can have a beginning of the decomposition, normal, then the condition change suddenly, and then we go through the mummification. Or vice versa, a body start mummifying, and then because a spillage of water, because the arrival of change of direction of a river, because a storm or whatever, the amount of water in the tissue will increase, and we can start again with the decomposition process. So when we study in the books, everything seems to be easy and unique. In the real life, in this case, in the real death, the conditions are not so clear and not so unique. So we can have the mix of the two. Third point, remember as well that a body into the water doesn't go through decomposition or through mummification. We will have what we call the saponification, the production of substances that are hydrated, and the shape of the body will be maintained. Of course, if this one is a normal pattern of decomposition, we have some insects that are associated with the different stages. The beginning of the story, flies. The end of the story, beetles and clothes moths. Why this? Because these kind of insects are adapted to feed on wet environment or on dry food. At the beginning, with certain chemical condition, at the end, we are the one. Anyway, if you look at all the processes, could be a decomposition from fresh to skeletonized, could be a mummification from fresh to mummified. What is changing from a physical point of view is the amount of water. We have a loss of water in a very evident way. This is the theoretical base. Now, if we focus our attention on the body, which kind of insect are we able to see on the body? On an archaeological context, we have insect associated with the body, that one I showed you a few seconds ago. We can have the ectoparasites. We can have insect associated with the offerings, insect associated with the environment, and in a museum collection we could have as well pest insect destroying our sample, what we call the museophagus insects. And unfortunately, I didn't have the time to put the title of an article that has been published yesterday, in which I describe, but I could pass you the title, in which I describe the way to distinguish in a museum collection between insects of the first 4 points and insects that are destroying our collection. In my talk, in the next cases that I will show you, I refer to Italy, this strange country in Southern Europe. In pink one, this is the flag, and what you know about Italy normally is this one. In Italy, we have Rome with the Colosseum. Rome is a fantastic city, but we have as well Venice. I come from Treviso, that is close to Venice, that is with the gondola. This fantastic picture gave you an idea about Venice. Then, of course, Italy has the best football team around the world. We are the creator of pizza, and this is a pizza margherita. And then we have invented as well the spaghetti. And I show you this very romantic cartoon that is Lily and the Vagabond, where they are eating spaghetti. Of course, spaghetti is not a dog food, it's for people. But so this is Italy. And what is the cultural background of Italy? The cultural background is based on the Roman Empire that was related as well with the Greek culture. And then in Italy, we have Rome. And in Rome, we have the city of Vatican, that is the core, the bulk of the Catholic Church. So we have from one side the Roman culture, and on the top of it, the Christianity in terms of religion, but as well in terms of culture. And then it's for this reason that combining these 2 points, we have a fantastic, a huge archaeological background. And if you dig somewhere, you are sure that you will find something of from an archaeological interest, or of archaeological interest. I start with this story because I become friend of these saints, friend in terms of because I work a lot on the remain of this guy, so I consider them a sort of friends. And in this case, we can see how in which way the insect can help in human identification. So the Catholic Church is trying to verify if the remain of the saint, or blessed in this case, belong indeed to that person. Because there is another thing that happened in Italy, but it happened as well in Spain and in other European countries. We had a lot of invasion, a lot of changes from a political point of view, a lot of wars, and sometimes these kind of wars affected as well the cemetery, and some churches were destroyed. People, I think, are stupid because we have to respect the religion and the culture of the other one. But in the past, unfortunately, as well in the present, is a common habit to destroy the tradition of the other one. It's for this reason that I told you that history is magistra vitae, but she doesn't have a lot of students. Anyway, in that case, the church, the bishop, asked to a group of scientists, and me, I was among them, to verify if this Bonaventura Carretti, that lived at the end of, in the 18th century, the bones that we have belong indeed to him. So we have some anthropologists that did the biological profile of the bones, and then they gave me the rest, what they call the dirty stuff. And on the top, on the left, you can see what I received. It was an amount, a mixed stuff of puparia with some head of flies that is telling me that the cycle happened inside the place where the body was. This is the genus Hydrotea capensis. That is a very common species in buried remain in crypts. I found as well Megazellia scalaris, that is another, is a coffin fly able to dig into the soil. This one was the Diptera part of the sample. No blowflies, no flesh flies, that's it. Then a huge amount of different beetles, some of them, in the case, the left one, the weevils, sorry, associate with the decomposition of wood, and on the right, Lagelandia anophthalma, that is a typical of wet environment. That means typical of an environment like this one, and that gave me an idea about where the body stay for a time. But at the same time, I found some remain of cloth moth, the cocoon on the left, on the upper part of the slide on the left, and then remain of some spider. They were quite strange because if the previous one were speaking about a wet environment, this one were speaking about a dry environment that could be a church, a crypt, or something sheltered and dry. This means that first information, or better, first interpretation of the insect community, the body was moved potentially from an environment like this one to a dry one. But looking among the sample, I found some piece of wood, and you can see here the holes performed by the weevils, and as well iron rust. here, that of course was related with a wooden coffin, a wood coffin of a very poor wood. But I found as well some brick fragments and other kind of mineral material that were related with something like that. So when I wrote my expertise, I sent to the bishop. I said, "Look, I was working blind. This body was maintained for a period in a wood coffin in a wet environment, and then I don't know what's happened, but it was moved in a sheltered environment, potentially a church or a crypt." And the bishop wrote me and say, "And the inset were able to tell you all this stuff?" I say, "Yes, of course." And he told me, "Yes, it is. It's exactly what's happened to the remain of this person, because between these two, we have this." I know that for you that you live in Asia is a quite strange situation, but in 1804, Napoleon, that he moved from France to the other European country, he made this law where he forbidden all the cemetery associated with churches or associated with city. So we had to move the cemetery from the city to outside the city. And sometimes they destroyed as well the cemetery. What's happened? After the death, after 20 years after the death of this guy, of this Carretti, the priest of the monastery, knowing about this law, asked to the other monk of the monastery to collect the bones of Bonaventura Carretti, because he was a saint, and to protect, to save them in a sheltered, protected place, and they shelter inside the church. And it was found years and years later. So the inset were able to tell exactly the story and confirming that that remain passed from an environment to the other one, and from an historical point of view. We know as well the reason now. Then I want to move to the First World. This is a group of Alpine soldiers. I've been an officer, lieutenant of this corps. They are soldiers that are expert of mountain war. So during the First War, we have a huge amount of missing people in Italy. We are speaking about 650,000 missing soldiers. And the member of the family are still hoping to have at least the remain of these people. For this reason, we start with a project. In this project, we are collecting what are called the militaria, this means fragment or element belonging to the soldier, but sometimes we find as well the skeleton. And the idea is to try to give the name to these people. We have, this is an Italian soldier, First War, that means we are at the beginning of the 20th century, 1990-15, 1990-18. The war was between the Austro-Hungarian Empire against French, Italian, British, American, Canadian, and Australian soldier. And this is what we can find. We have the numerus, we have the femora, helmets. We can say that it was an Italian soldier because the shape of the helmets. We study as well a little bit the algae and the fungi, IFI, present on the remain, but they were not informative. So what we discovered was a huge amount of puparia among the ammunition pockets. Which kind of flies? Lucky they, because we were able to identify the puparia. You know, the puparia are not easy to be identified, but in this case we were able to do. I was able to do, and I found Protoformia terranove, that is a first colonizer of an exposed body. Then I found as well Formia regina. I know that the American and the British people, they say Regina, but the Latin name is Regina. My apologies. And that is another colonizer of the body at the beginning of the story. And then as well, some puparia of Fanide in the genus Fania. These ones are used to come later and are able to dig as well. So what the insect were telling me? They were telling me that the colonization potentially started at the end of the spring, maybe beginning of summer, just because of Protoformia terranove in that part in northern Italy is active indeed at the end of the spring and at the beginning of the autumn. Then during the summer disappear. In contrast, Formia regina is active along the whole period, and the Fania potentially canicularis is present only in the summer. So to have the wave of colonization, I have to expect this pattern and not this pattern from here to the next year. Then there is another detail that it comes from the uniform, the fragment of the uniform we found on the remains. We found them later that confirm this first hypothesis. But what we can say in addition about this case, if you remember, I remind you, going back, the body was found under the soil. It was a more or less 60 centimeters of soil between the walking layer and the position of the body. This means that something happened and some soil covered the body. Usually, during the war, the camerate are used to recover the death. Why they didn't recover this guy? And what's happened to him? Okay, we saw from a phenological point of view, so we can think that he died at that time. But then, if we look at the behavior of the flies, we have that the first two are able to colonize a body only if it's exposed. The third one is able to colonize a body that is buried. This is a canon. Look at this hole. This hole is the result of a bomb during the First War. And now check the size of the two soldiers. So the amount of soil that was moved by the bomb was huge. So what we can think, this guy was attacking the enemy. He was shot down, or better, a grenade explodes close to him, and the school was injured and he died. The enemy attacked the Italian forces, and they reached the Italian trenches. So this guy, the body of this guy, remained behind. No one of his friends was able to collect him. After a couple of months, the Italian artillery start bombing to recover the position. In these 2 months, we had the colonization of Formia regina and Protoformia terranove. Bombing, something happened and a huge amount of soil recovered the remain of the soldier, and he stayed there for a century. In fact, during that war, during that battle, the Italians were able to recolonize the position, but they didn't collect the body of this guy because it was just under the soil. They didn't find him. Why we are working in this direction? Because as a project, we are trying to give back to the family the remain of the soldier. So insect, in this case, to understand the season, or better, the time of the year when they died, to short listening the missing people list, and at that point it will be easier to perform all the DNA analysis or whatever, and at that point we will be able to give a name to these people. We cannot perform the DNA analysis on 600,000 people and 600,000 families, and at the moment we don't know how many members of the family are still in life, because 1 century passed. So anyway, we are working on that direction. So looking at the future, about this point, we need to break the border, and I'm quite happy because the husband of my sister is from Austria, that 1 century ago was an enemy country, and now we are friends. And if you look at the faces of these people, this one are Italian, this one are Austrian, they are exactly the same. So we need to stop the very stupid war that we are around the world. We move to the last case that I want to present, because the time, and is from Castelsardo. Castelsardo, okay, we go back to Italy. This is Italy, again, it's in Europe. As you can see, we have water everywhere, and we have as well 2 big islands, this one and this one, because the third one belongs to France. The name is Corsica, that is French territory. This is Sardinia, okay, and this one, this triangle, is Sicily. Okay, we go to Sardinia, and we go on this fantastic place that is called Castelsardo. You can see from Google Map the position of the village, sorry, and this is the church. Working in a restoration, they were restoring the church, and under the church they found a crypt with these 2 bodies that they call, as well in a romantic way, Bob and Mary. They were not related. It's just because the coffin, they collapse one on the other one, and we start working on them to understand what's happened. And what we discovered, what the insect were able to tell us, is something amazing, because I show you the list of the species. We found Callifera vicina. Please do not say vicina, it's vicina. And we were able to identify the puparia. Then, of course, some sarcophaga, so flesh flies. Hydrotea capensis, again, very common in crypts. And then this one, again, Formia regina. But what happened? When I found this species in this remain, and we are speaking about the 19th century, 18th, 19th century, I check about the distribution of Formia regina. And Formia regina, in the fauna of Europe, that is the database about the distribution of the species, is not present in Sardinia. It's not present in Sicily as well. But in the 18th, 19th century, it was, because I have the evidence. I have the puparia that are telling me that that species was present in that place. I know that I have 1 minute, more or less. What mean? This means that this species was present in Sardinia 200 years ago, and this one was related because the trade between the city of Genoa, where I live now, that it was a very important city, and Sardinia. So they were trading not only food, goods, whatever was they were able to sell, but they were moving as well insects, as we are doing nowadays, as we did in the past, as happened with the Roman. But anyway, we have the evidence that in the past as well, that species was present, at least in this part of Sardinia. Nowadays, it's completely extinct. It doesn't exist anymore. Last but not least, on this case, we found on the remain 4 different species of flea, and one of them is related with human Pulex irritans, can be related with mice and rats as well, but is as well an ectoparasite of humans. And a collaborator of us, that is a dermatologist, analyzed the whole bodies of these 2, Bob and Mary, and what he found is this fantastic stuff that is the evidence of a bite of flea on the skin of one of the 2 mummies. This means that when they were alive, they have the fleas on them, and now we are looking for the pathogens that could be transmitted by the fleas. What we are doing is something that has been already done for lice by Didier Raoult, that is one of the most important microbiologists around the world, despite his position nowadays, that he was able to extract the DNA of Bartonella quintana from lice and the teeth of these remains that belong to the Napoleon Grand Army in Vilnius. I think, tell me if my time is over. Yes, it is.
Yeah, Dr. Vanin, we have 5 minutes more.
For question.
But well,
okay.
If you want to finish,
okay.
Proceed.
As you can see, I have millions of pictures. I don't want to go through all of them. I would like only to tell you that my lab, the name of my lab is FLEA, just because the FLEA I'm used to find in the archaeological context, and FLEA mean for me Forensic Laboratory for Entomology and Archaeology. If you want to come here to work with archaeological stuff, as you can see, there are millions of things to see. And thank you indeed for your attention, and I hope you are still alive and not sleeping at the moment.
Thank you, Dr. Vanin, for your impressive presentation. And now we will proceed with the Q&A session. For those who want to ask some question to Dr. Vanin directly, please do so by turning your mic on. We always welcome an interactive session. Anyway, if there is any question posted in the chat room, I will read them. So, if there is no question.
Hi. Hi, doctor. I would like to just know, like, I'm a forensic anthropologist, so, but I do excavation and all. I just wanted to know, like, how do you think archaeo-entomology helped in bringing back those soldiers from the First World War? Like, because obviously on the perspective of anthropology, when you look up at bones, we try to create the biological profile and all. But because it's been like so long ago, so how do you think entomology like fits into this?
So the aim of the entomology is to find the season of the death, and this is important because we know that different regiments, different troops come in different times. Just to give an example, the 7th Regiment of Alpine Soldier was fighting in that part only from April till May. Then they rest, and another regiment arrived during the summer. Because we have the list of the missing people of each regiment, if I know that the guy died during the spring, I can focus attention only on that regiment. Second point, I use anthropology to try to estimate the age of the guy. I try to check if it belonged to the privates, the soldier, or if it was an officer. And we can see or by some element of the uniform, or for example, because it had some rich chirurgical intervention on the teeth. If we can find some gold on the teeth, it was not a soldier, it was a member of the higher part of the army. Okay, so the combination between entomologists is telling you, look, you have to focus the attention only on that troops, not on the whole Italian army. And then you implement with the anthropological analysis, really you can narrow and narrow and narrow the list of the potential victims. At that point, you will not have to check among 650,000 missing family, the family of them, but maybe 10 people, 5 people. At that point, it would be easier to give a name. We gave the name to some of them, so we are very happy from an ethical point of view.
Perfect. Means basically just narrow down to the people.
Missing people list.
Yeah, sure. Thank you, doctor.
Welcome.
Thank you so much for your answers. Any question more? Well, if anyone ask, I will ask some curious thing that I have on mind. I would like to know if you find in on some of your cases Syndesomia nudiceta.
Sorry?
Syndesomia nudiceta, if you found in the...
No, so this is a nice story. You know that I published about that species. That species arrived in Italy only a few years ago. Mm-hmm. And so from an archaeological point of view, no, absolutely. I will be happy to find them because that means it's a record of the past. It's what's happened, for example, with the black soldier fly, Hermetia lugens. What we think is that it appeared in Europe in the 50s, during in the period of the Second War. But an Italian guy that is a friend of mine, Benelli, he discovered a specimen in a coffin of the 16th century. So there is a specimen of the black soldier fly that date 400 years ago in Italy. So we can say that maybe it arrived, the weather was not so nice, so it disappeared. With Syntensomia nudiceta, what I can tell you is that it arrived in Italy a few years ago. I performed the molecular analysis, and my population here in Genova is not related with the Spanish from a molecular point of view, but is related with the Chinese one. And if you look, the Genoa port is the second port in Italy for trades, and the main so the majority of the material the ships, come from China. So that means that it traveled from China to Italy in a container or somewhere like that, and then it colonized Genoa. But what is amazing, that at the moment, because I perform a sampling that started 2 years ago and is still going, is present only in Genoa, not in the other port close to Genoa. That is explaining me a movement from the sea and not from the land. That could be one of the hypotheses, because you know better than me, Spain is not so far from where I am. Mm-hmm. We have only a little bit of France between us, but it didn't come from the land. It's coming from the sea.
Okay, thank you so much. In the chat box, they ask about your email for contact you.
My email? My email is at the end of the presentation. So anyway, if you type Stefano Vanin on Google, you will find me. Google is a sort of a fantastic library where you can find whatever you want. But I share just 1 second the slide so you can have the mail. Voila. Voila. Can you see the mail?
Not yet.
Not yet?
Wait. Yeah, it's now.
Okay.
I will type your email in the chat box.
Okay, anyway, Gmail is the best one, because sometimes the university one is full of mail of student panicking about exam, whatever. So Gmail, stefano.vanin, gmail.com is the best one. Anyway, if in the future, if you want to start working in funerary archaeo-entomology in your country, indeed, I will be very happy to help, to provide what I know. Of course, I have a huge amount of things that I would like to know, but whatever I know, I'm happy to share. This is another point. When you work in this domain, you have to start considering the context where you work. There are some places, because the culture, because the religion, where the body are mummified or are preserved or are considered in a certain way. In other religion, in other part of the world, the body are considered in another way. So what I suggest all the time is to respect the culture of the other one. If in a country the body of a dead person cannot be touched, do not push to do an investigation in that direction. First, I told you respect. Second one, our curiosity. Then there are other aspects. They are more related with forensic, with the distribution of disease, where we have to push the culture, say, stop. In this case, it's important to do this because we could save life of other people. At that point, we need to discuss a little bit with the tradition, with the religion, with the culture, to save life. But if not, if life of other people is not cannot be improved about our study, please, the respect is the first point. I will never ask in a country where I know that the body are protected to make my analysis. Absolutely not. So please start considering where you are, what is the habit in your country. If doing something you can affect the sensibility of someone, if the answer would be the thing is important, I'm not touching the sensibility of anyone, I can improve the science, great, I do. If not, there are other parts of the world where you can do your research.
Thank you so much again, Dr. Vanin. Any question more from the participants?
Well, I'd like to ask a question to Dr. Vanin. Thank you very much for your amazing presentation. Definitely, I'd like to visit Flea Lab one day in future. Hopefully,
When you want,
after the pandemic, yeah. Right. So from your presentation just now, you mentioned that the Carifolia vicina was not found in Sardinia, right? But it was found many years ago, like, and then you mentioned about the extinction. So what, in your mind, in your opinion, what contributed to the extinction of the blowflies? And should we concern or aware about the extinction of blowflies? Thank you.
So what is extinct is not California vicina, but is Formia regina.
Formia regina, all right, thanks. Yeah.
Formia regina. One of the hypotheses I was doing is related with the relationship between human, animals, and these flies. In the past, we were used, in Italy at least, to live very close with the animal. So the house was different rooms. In some of that rooms, you had animals inside because the weather, the temperature, or whatever. So the relationship between human and animal were very close. With the time, the situation changed, and then we have the separation between human and the other animals. And we know that Formia regina can be related with animals. This is the first point. The second one is related with the way in which the species colonize an environment, is what happened, for example, with the Syntenisomia. The species, some specimens arrive. We know that they arrive.
Yeah.
But in a certain period, the environmental conditions are below the survival threshold. The threshold could be the temperature. So maybe that in the past, in Sardinia, the temperature was too high or too low to allow the survival of the species. In that specific crypt, it found the condition to survive. And look, I show you the picture of the Castelsardo is a port. So maybe it arrived, it came down from the ship, It was able to colonize a little bit the environment, but it was not able to colonize the whole environment because of the environmental conditions. With the example of the Synthenisomia, what we had that the Synthenisomia survive only when the temperature, the isotherm, that means the average of the temperature during the year, is 20 degrees. In Genoa in English, Genoa in the Italian world, this temperature was reached because the global warming exists, okay? Was reached only a few years ago, because before the temperature was around 18. So now when the temperature reaches the threshold, the fly is able to colonize. Maybe that, I hope no, the temperature will increase too much, this species could disappear because it's too warm.
Yep.
So we have this combination, I think, for the blowfly, the association human-animals, the way in which we manage the environment and the relationship with the other species, and as well the environmental condition.
Yeah. Thank you so much for your explanation. Yeah, it's great to know. Yeah, the environmental factor and the human behavior that can impact the existence of the flies. Yeah. Okay, thanks.
Thank you for the question.
Thank you so much again.
Anyway,
any question? Sorry, doctor. Any question more?
I kill all the audience, so my apologies. Anyway, Dr. Ching, if you will come here, I will be very happy, not only to show you the flea lab, but as well going around Italy to have some nice food, nice wine, and enjoy as well in collecting flies if you want.
Yeah, perfect. That is yeah. I really love to visit one day. Yeah, thanks for that. Yeah, the same thing. If you really hope after the pandemic, you will have a chance to visit my lab, which is called the F.U.N lab, Forensic Urban Nutritional Lab. Yeah, in Malaysia, and enjoy the warm climate here. A warm welcome.
Okay, so if there is no more question, thank you, Dr. Vanin, for your answers. Please allow me to close this seminar now due to the time constraint, and don't forget to sign the attendance form to get the e-certificate. For those who are interested to join us in the next webinar series, the date is set on 23rd November by Dr. Paola Magni from Murdoch University in Australia. I'm looking forward to meet all of you again in our next webinar. Again, I would like to thank Dr. Vanin for agreeing to become our guest speaker for today, and the participants for joining us, and remember to stay safe. Goodbye, everyone.
Bye-bye.
Okay, goodbye, Dr. Vanin. Yeah, take care, stay safe. Yeah, see you. Take care. Bye-bye. Everyone, thanks. Yeah.